The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
This month, how tiny termites know how to build massive nest structures, an initiative to keep tabs on infectious causes of livestock abortion in Africa, and how weight gain shifts the balance of food-related memories. Hello, this is episode 93 of the eLife podcast. I'm Chris Smith from The Naked Scientists. Up first this month, scientists have suspected for a long time that one of the drivers of emerging diseases, particularly in the tropics, is the conflict between our human activities and nature. Where we encroach, there are opportunities for zoonotic, in other words, animal-borne infections, to jump the species barrier. But what are the risk factors that make this more likely to happen? One form of malaria, called Knowles eye, that's normally carried by monkeys but occasionally does jump into people and is now increasing its incidence across Asia, has provided Amelia Johnson from the University of Glasgow with an opportunity to study why this might be happening.
Everyone's familiar with malaria between humans, so human to human, passed with mosquitoes, a big problem in Africa still. But in Malaysia, there's been huge reductions of malaria. So they've managed to nearly eliminate it using bed nets and public health controls. And at the same time, there's been this huge rise in monkey malaria, a different type of malaria where you have malaria circulating in non-human primates and in particular communities which are interacting with the edges of the forest, you start to get spillovers. The mosquitoes that are biting the monkeys also bite the humans and you start to get malaria that way.
And so what was the unknown or the question you were trying to probe?
This particular type of zoonotic malaria in humans, we knew that was associated with deforestation and with communities that are living and working at these forest edges. But what we didn't really know was anything about the malaria in the monkeys themselves. You can imagine that if monkey populations have really high rates of malaria, you're going to get a lot of spillover into the humans, whereas if it's really low, maybe you won't see as much. And actually, all the literature had been assuming that all monkeys across Southeast Asia had really high rates. So we wanted to start to untangle whether that was true and also what might be driving that. So if you understand what's driving that, maybe you can start to understand where the risk factors are for people.
So what was your approach then? How did you begin to try and unpick those questions?
Firstly, it was a meta-analysis. So we took all of the literature that we could find, all of the studies where they had collected primates, sort of mainly macaques, anywhere in Southeast Asia, and where they'd tested them for malaria, what those results were. And we pulled them all together and looked across different regions and said, okay, across all of the monkeys, so it's about 6,000 monkeys in 150 different locations, how many monkeys are infected? And then the next thing to do was to look at in the places where there's lots of infection versus the places where there isn't really any. What's the difference? Is it being driven by deforestation? Is it being driven by where there's more forest or less forest, or where there's more humans or less humans?
And did you see some obvious drivers?
For other infectious diseases in other wildlife hosts, there have already been some links between infection and deforestation, and that's exactly what we saw here. So in areas that are really fragmented, where you chop up the forest into smaller patches, you have higher rates of primate malaria in the non-human primates. And we think this is possibly because where you have a chopped up forest, you have a lot of these forest edges, and that's where the mosquitoes like to live. So where you have more mosquitoes, they're probably passing malaria more readily between the primates. The other thing we found was that it's also associated to where there is more forest. So not only is the forest more chopped up, but there's more of it. So actually there's a bit of a trade-off. You need an ideal amount of forest and fragmentation in order to get these areas of high prevalence in the wildlife.
And as well as the mosquitoes being attracted to the margins, are the primates also tended to move towards the margins as well? So it brings a concentration of both the vector, the mosquito, and the animals that can be the source and also be the target of the infection.
Yeah, that's exactly right. So the main species that we found had been studied are macaques. And these are really highly attracted to areas where humans live and work. Typically, they're nuisance animals because they love to eat leftover food and be in and around villages. So they are quite often living in these margins between urban areas and forested areas. So that's exactly right, where you have more fragmentation. You have both the primates and you have the mosquito vectors, and those are both obviously closer to the humans.
Often, when we uncover a mechanism like this, it also inevitably leads us to some solutions or some interventions that can try to reduce risk and so on. So, does this point towards some obvious directions we should be moving in? Obviously, fewer people, less fragmentation would be ideal, but that's hard to achieve. So, what do you think we can do off the back of what you've found?
It doesn't necessarily lend itself to a clear solution, but what it does say is that it is absolutely essential to consider both wildlife compartments and landscape composition in public health measures. So, this idea of One Health or planetary health, everything is so interconnected and so entangled that you can't consider public health without the environment and the wildlife aspects. There is no silver bullet to this. With human malaria, you can use bed nets, but because this is more about how people are interacting during the day in their working life at these forest fringes, that's not applicable here. So, what we need is solutions that are community driven and sustainable and more to do with landscape management in order to reduce risk.
Amelia Johnson there. Have you ever heard of stigmergy? No, I hadn't either. The dictionary definition, though, is the trace left in the environment by an individual action that stimulates the performance of a succeeding action by the same or a different agent. And this is how Giulio Facchini from CNRS in Paris thinks termites, despite measuring just millimetres in length, can work together in their thousands or more to construct massive, intricate nests. He gave them coloured pellets of mud to work with to make it simple to follow their progress, and fashioned some initial curved shapes into his study arena. These shapes motivated the termites to assemble the coloured mud into very specific structures. In other words, by building a certain shape, other workers who encounter that shape are guided as to where to put the next piece.
Termites, like other insects that work collectively, interact according to a principle that is called stigmergy, which simply means that each individual doesn't know the global plan of their activity. That could be construction, that could be foraging, whatever action. They don't know the full plan, but every single action of an individual leave a trace for the following one to take on the work. And so we know this, but there is no common agreement on what should be this kind of trace. And there are a lot of different ideas in the literature. That could be an odor, that could be a gas concentration, that could be some touching, feeling. So we wanted to explore what this trace could be.
One example, for instance, these animals build big, complicated nests that they've all worked together to construct that architecture. Mm-hmm. So how would they all know what to do and where to go in order to do that? Where are the cues coming from? I suppose that's part and parcel of this, isn't it?
Yeah, for example, ants can leave some odor within the small piece of mud that they add to a construction to inform the following ants that they should add more mud at the same place. And that can lead to the formation of pillars. The same thing was proposed for termites, but actually we showed that odors in this case are not necessary and information can be transmitted just through the shape of the environment.
So talk us through how you reached that conclusion then. What did you actually do to disprove the pheromone or chemical marking idea and come up with the idea that it's all to do with the shape of what's already there?
The idea was to prepare some topography. Instead of having, for example, a flat environment where termites move, you add some bumps, some details, curvature. So we prepare this environment with mud that has been sterilized in the oven. So we are sure that there is no odor in that, there is no like chemical traces. And we observe that there will be some organization in the building actions, even without this odor.
So how did you track that? Was that sort of you video a bunch of termites interacting with that environment you've created to see how the shape of the environment changes the behavior of the insect?
Okay, so the idea was quite simple, but quite tricky at the same time. The first thing is that if you leave termites with mud, at some point they will dig and prepare some small balls of mud that they use to build things. So the idea was to leave termites prepare these pellets in some preliminary experiments. For that, we use some gray mud. And then we collect these pellets, we put them in an oven, so we sterilize them, and then we add that to a new experiment where all the environment, the one with shapes that I told you before, is prepared with the same mud but of a different color, so red. And so this way, when we are filming the system, we can actually separate the pellets from the environment. And for example, when a pellet is taken out, we can do what is called the differences between images, and we will see a dark trace where a pellet has been taken out and a light trace when a pellet has been deposited, which give us information about the building actions.
And because the pellets have been baked in the oven, you reason this has driven off all the chemical cues. So the only thing left that could be guiding the deposition and the removal and the movement is the shape.
Yes, exactly. And we, in order to be more sure about that, what we prepare is a flat disc on which we add just two bumps, which are of the size a bit larger than termites. And then we observe that the pellets that have been placed were all over the disc were moved mainly to the top of the more curved region, which suggested that this shape can guide termites in their building actions.
A couple of things obviously spring to mind off the back of that. One is, how do the termites gauge the shape and what sort of shape are they interested in? And second, how do you then rule out that having gauged the shape and begun construction, they don't then deposit some kind of chemical cue or reinforcement that then encourages other termites to carry on doing this?
Two very good questions that we try to address. So the first one, actually, the answer came from a kind of surprise that we had in our experiment, because I told you that we observed an increase of pellet deposition on the top of these pillars that are more curved, but we also observed the same kind of increase in deposition at the edge of the disc, of the flat disc, a region which is not very curved, which is pretty flat. This was a surprise for us because we had both a flat region and a curved region. But in the end, what we understood is that what is in common between this tip of pillars and edge of the disc is the fact that the evaporation is very strong there. And it turns out that whenever you have a curved region, which is humid, evaporation is stronger there. So what actually happens is that both the edge of the disc and the tip of the pillars are region where, thanks to this specific shape, evaporation is stronger. And we believe that the way termites sense this shape is precisely through evaporation flux. And this is quite well accepted in the community because termites tremendously need humidity to survive. So they must be able to sense where you switch from a dry to humid environment, which is exactly where evaporation is strong. And so concerning your second question, we cannot actually rule out the fact that once the building process is started, some pheromones, some specific odor laid by termites could also play a role in the construction. So, and we say that in the paper, this mechanism could be like on the top of the shape guiding, there could be pheromones guiding, but what we say is that at the very beginning there is no pheromones, so there is no need for that to start, which is something new because no one has suggested that before.
Amazing piece of work that. And Julia went on to tell me actually that this process of stigmergy is almost certainly just one from a range of cues and stimuli that the animals use, but it's clearly a very significant one. You're listening to the eLife podcast with me, Chris Smith. On the way, the challenges to overcome in understanding neurodiversity. Before that though, lack of reliable data on the etiology of livestock diseases, especially in Africa, is a major factor constraining the design of effective livestock health interventions to improve livelihoods, food security and public health. That's how Felix Lancaster from Washington State University opens his paper, in which he's worked with government livestock field officers and farmers in Tanzania to follow up cases of animal abortion and help to safeguard human health and livelihoods in the process.
So this study took place in northern Tanzania, near a town called Arusha, just south of the Kenya border. We teamed up with what are called livestock field officers. They're not veterinarians, but they're like veterinary assistants. There's one of those typically for every 4 or 5 villages. And so we asked them to let the villagers know that if they had an abortion in their livestock, that they should contact the livestock field officer using their mobile phone. That triggered an investigation that allowed us to collect samples and determine what was causing abortion.
So what samples did you go and collect?
If the fetus from the abortion was still present, we would collect some tissue from the fetus and from the placenta and the fetal membranes. We also collected blood samples and vaginal swabs from the dam, from the mother.
And what turned up once you began to collect these, both reports, but also in some cases get these specimens? Was there a clear picture emerging?
The first picture that emerged was that abortion was relatively common. So across 15 villages in a 2-year program, we had over 200 cases of abortion. Of those, we were able to identify an infectious disease cause in just over 40 of the cases. So first thing is that there's quite a lot of loss in livestock caused by abortion. And secondarily, a relatively large proportion of these are caused by pathogens, some of which can be controlled with vaccination programs or other animal health programs that aim to stop the transmission of these pathogens.
Were there any hotspots?
So we looked at the number of cases reported per livestock field officer, and there was a great variety of cases. 2 of the livestock field officers didn't report a single case out of the 15, and one of them, who reported the most, reported 84 cases. And so it appears that some villages had a lot more cases than others. We weren't able to determine whether that was because those livestock field officers were just particularly more active, or alternatively, whether actually, as you say, there was a hotspot in that village and there were more cases being reported.
What about human contact? Was there any data collected on exposure of humans to some of these things so we can get some idea of, or some insight into the potential conduit that they might pose to infection spreading into people?
Yeah, so in 61 of the 77 abortion cases where a pathogen was detected, it was a zoonotic pathogen, so a pathogen that can infect humans. And in many of these cases, a human had assisted with the delivery of the abortion case. In nearly a quarter of the cases, somebody had helped deliver the abortion and as a result would have been exposed. And these are highly infectious tissues. And so these people who've assisted with the abortion would have been at risk of being infected with a zoonotic pathogen such as brucella or coxiella or toxoplasma or Rift Valley fever.
So what are the take-home messages then? What are the implications of what you've found in terms of next steps?
There were a number of different pathogens that were detected, like Rift Valley fever and Toxoplasma gondii and Coxiella burnetii and Neospora caninum. And some of these pathogens have a particular transmission pathway, and so we can advise livestock keepers about reducing those transmission risks. For example, Rift Valley fever virus we detected in the milk of the dam. And so this indicates that drinking unboiled milk is a potential source of infection in humans. Similarly, we found Neospora caninum, which is a pathogen of dogs, but can cause abortion in livestock. Again, there are areas of health advice that we can give to the livestock keepers regarding reducing the likelihood of transmission of this pathogen from dogs to cattle. In addition, we found that we were able to detect the pathogens simply using a vaginal swab from the dam. That was really an interesting finding because it showed that quite simple, relatively cheap, and easily accessible samples are able to produce a lot of data on the causes of abortion. Fetal samples and fetal tissue samples were collected, but often those tissues aren't available because local dogs eat them, or predators eat them, or they get thrown away by the livestock keepers. And so these tissues are not always available to sample. So in these situations, we've shown that there is still a very useful source of samples, and that's simply collecting a very easily obtainable vaginal swab from the dam, and that on its own is often sufficient to produce an informative result.
Thanks very much to Felix Lancaster. The term neurodiversity owes its origins to the Australian sociologist Judy Singer, who proposed it in her 1998 master's thesis, Odd People In. There, she's highlighting that since everyone's brain develops in a unique way, no two brains are ever the same, and so there's no real meaningful definition of normal when it comes to what's inside our heads. But a quarter of a century later, the language and the understanding of this concept are still lagging. And this, Jola Lainka and her colleagues at the University of Bath argue, is retarding the crucial interdisciplinary collaborations between genetics, psychiatry and the social sciences and other fields of research that are much needed to understand conditions like autism, ADHD and dyslexia.
I find that the easiest way to explain neurodiversity is to think of biodiversity. It's one of the first things you learn in biology class, right? It's about the variation of all living things. So in terms of neurodiversity, it's that unique and complex variation in the way we think and neurological functioning. So it relates to conditions like autism and ADHD because we all have these autistic or ADHD traits in different degrees, leading to very diverse ways of thinking. So those who are diagnosed with these conditions are just on the extreme higher end of this natural distribution. So neurodiversity just encompasses this variation in thinking as a whole.
Indeed, because we do come around to the idea that really everybody has a range of different traits, don't we?
Exactly.
And it's more kind of how many or to what extent those traits are manifest that give you your degree of neurodiversity.
Exactly that.
Is that the point you're making here, that previously we've tended to put conditions in boxes?
Exactly. So that's where you find that categorical approach where if you have this number of traits, then you are autistic. And if you have this specific number of traits, then you have ADHD. But actually, what we find is these traits are overlapping with each other. So using that neurodiversity approach, we can shift towards more of the dimensional approach where we can measure these traits as the spectrum that they are.
Does the language we use, though, support that kind of approach? Because where we have historically adopted a kind of a diagnosis base, you have this condition and this condition, rather than you have this range of traits. If the language isn't there to define it, then scientists can sometimes struggle to agree on what they're actually saying.
Exactly, which is why we kind of need to move slightly away from this deficit medicalization approach to using more inclusive language that's not just focused on this deficits, so that we can really portray this dimensional way of thinking in terms of neurodiversity, in terms of these neurodevelopmental conditions.
But do scientists actually agree on what they are studying? In terms of, if you are studying autism, does one person's definition of I'm studying autism agree with another who might be interested in some of these trait-based approaches? And actually you've got lots of people working on the same thing and they don't realise it.
Exactly that. Absolutely not. There is no standardization. The clarity of the terms is actually one of the difficulties. in this field. So we need to come together across disciplines to have this agreement where we can reduce all these overlapping terms that are used to describe the same things. Like you said, we have a lot of people actually investigating the same or similar things, but they're not aware of it. It shouldn't really be that as difficult as it is. So this is why that interdisciplinary work and clarity between terms, working with each other and communicating with each other better, needs to happen.
And is it a relatively easy landscape to get funded in this space?
In terms of funding, there's actually a lot of variation in terms of which disciplines get funded more so than others. So you see a lot of funding towards genetic or neuroscience research, where it's about finding targets for personalized interventions and finding ways to further the intervention a lot later down the line. But a lot of people from these neurodiverse communities are actually wanting to see improvements on their day-to-day lives. They're wanting to see improvements to programs that are supporting them now. So it's about redirecting some of this funding to promote that, not necessarily stopping any of the great work that's happening, just creating a bit more balance.
So what's the ultimate goal here? If I was talking to you in 10 years' time and everything you've written here comes to pass and does it in record time, what do you hope the shape of the conversation we'll be having will be? What do you hope will have happened?
Well, one, that we agree more on the terms that we are using. We include neurodiverse communities a lot more so that we are actually researching things that are important to them as well. And the communication between researchers, between disciplines, has improved. So there's a lot more interdisciplinary work that's actually being conducted, and the communication between those communities has improved. Things like patient and public involvement, co-designing studies with people from these communities, and things like that.
Jola Lainka. The World Obesity Federation predicts that within the next decade, half the world's population will be overweight or obese. And all the evidence points to this being a one-way street. Once people put on weight, it becomes very hard to shift. One reason might be the way the brain makes food-related memories. Henrike Scholz from the University of Cologne has been looking at this using flies. Their brains are wired for food in the same way that owls are, making them excellent study subjects. And the crux of her discovery is that lean people and flies make less persistent food-related memories than fatter individuals, which also tend to attach a reduced reward to food consumption. Hey presto, the toxic combination of an enduring impression that food packs a lower calorie punch than it really does. And that adds up to overeating and long-term weight gain.
When animals or humans eat or feed, the energy contents is measured. The insulin levels in the blood tell the brain how much they have eaten, and if they have eaten enough, they stop eating. This system is conserved between humans and the genetic model system we are working with, the flies, and we use the flies as a model to understand the behavior in humans.
So basically our bodies are counting the calories that we take in, and then they're storing a memory snapshot of when I eat this, I get this much calorie reward.
Yes.
So how does dieting or weight gain and weight loss fit into that then?
So it depends on your physical condition. So if you're a lean person and have periods where you don't eat, you think about the food, you then might eat the food, and the food is so rewarding that you lose the memory. However, an obese person goes through this phase of starvation. The memory gets much more stable. And if this obese person then eats food, the food is not perceived as rewarding. And the obese person still feels hungry. And that despite the fact that the energy level is full, and that results in overeating.
So if we unpick that a bit, a person who is overweight, they have a memory that when they eat certain calories, they don't get as much of a calorie reward as, say, a leaner person has for that food. The person who's overweight then attempts to lose weight. They go on a diet. So how does that affect that memory in the first instance? What does that do to the memory?
So the memory is much more stable. So there are different phases of memory. You can have a very short-lasting memory and a long-lasting memory. and these memories have different stabilities. In obese people, this longer lasting form of memory is much more stable. And if they eat, they don't lose the memory.
And is that memory one telling them, eat this, it's good? Or is it a memory that says, eat this, but it doesn't give you many calories, and that's what encourages overeating? Which way round is it?
So it's not about that the body is not measuring the energy content. It's about the memory, which is basically giving them the feeling that the food was not rewarding or not enough for them.
They've basically stored a memory that the food has fewer calories in it than it really does for them, and that memory is entrenched. It doesn't shake off very easily.
Yes.
And they weren't obviously overweight to start with, so were they always making memories that way, and then the memories become a problem because they make them gain weight? Or does the pattern of memory formation change with weight gain?
So the pattern of memory formation changes due to increasing body weight. So when you are lean and you're starved, you form a different form of memory than if you have accumulated body weight, basically. Then you form a different form of food memory.
So why is it not working in reverse? If you then shed the calories, why do you not make the lean form of the memory again and then lose the weight?
Because the organism adapted to this kind of memory because it's so stable, it is not erased.
And how did you put this to the test in the flies?
So you can basically generate genetically modified organisms which have different increases in body weight, and then ask how good they remember food-related cues and how long the memory lasts. And then in turn, how these memory influence their future food intake.
So you overfeed a fly, so you get a sort of a fly with a beer belly, if you like, and it lays down memories that are very long-lived, that when it eats this sort of food, it doesn't return much value, so it therefore tends to overeat.
Yes.
You mentioned that it's tied up to insulin, and insulin encourages us to store calories. So does that mean it's purely linked to carbohydrates, this then? And is the fact that our modern day diets are so dominated by sugars, people have often made low-fat versions of foods by substituting sugars instead of some of the fats, for example. Is that then at the heart of why so many people across the world now, more than half the world population, is dangerously overweight?
So we linked it to carbohydrates because carbohydrates are enriched in the Western diets, and this mechanism is triggered by carbohydrates, and it is extended to other foods. So, for example, these obese flies were deprived of carbohydrates, but they also overeat protein-rich food. So basically, this mechanism triggers overeating, and it's independent of what kind of food it is.
What are the implications then? Now you've got this association and you've unpicked how these food-related memories work, that must point towards possible interventions.
So the idea would be that, so one thing is that these neurotransmitters, which normally suppress the memory, could be basically activated in overweight people to block this mechanism or this formation of long-term memory when they basically go through a diet.
Food for thought. Henrika Scholz there. Well, that's it for this month. You've been listening to the eLife podcast that's produced by The Naked Scientists. There are previous editions, the references and the full text transcripts for these programmes, as well as details on how you can subscribe to this podcast. That's at nakedscientist.com/eLife. As well as details on how you can subscribe, assuming you don't already, to this podcast at nakedscientist.com/eLife. The Naked Scientists also publish our weekly science programme that covers the latest leading science stories. You can get that at nakedscientist.com/podcast. I will be back next month with another look inside eLife. Until then, from me, Chris Smith, thank you very much for listening. And until next time, goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.